So welcome everybody to another interview for the PHOENIX Open Innovation Testbed. PHOENIX-OITB is an EU-funded project bringing together service providers in nanopharmaceutical development and offering a single entry point to a variety of services in this field. Today we're looking at our fourth demo case. Here we have Elizabeth González Mira from the Institute for Material Science of Barcelona, which belongs to the Spanish National Research Council, as well as Alba Cordova from Nanomol Technologies. And unfortunately, our third partner, Leanbio, could not have anybody present today, but these three companies have all been working together on our demo case 4. They're all based in Barcelona and have been working on a NanoGLA formulation to treat Fabry disease. So they're going to tell us more about this demo case today. Elizabeth, can you give us a little summary of what you were doing in this demo case?
So thank you very much for the introduction. In this PHOENIX demo case, the Institute of Material Science of Barcelona, together with Nanomol Technologies and Leanbio, have joined efforts to advance the development of a novel nanomedicine for the treatment of Fabry disease, which is a rare genetic disorder. To put in context, this Fabry disease is caused by the lack or malfunction of a protein in the cells, specifically an enzyme called α-galactosidase, or from now on GLA. And this provokes the accumulation of fatty substances in the cells throughout the body, leading to the dysfunction of key organs and therefore to many clinical symptoms and can cause premature death. What makes our approach special is that we use the nanotechnology, specifically nanoliposomes that are well-known lipid nanovesicles, to encapsulate, transport, and deliver the missing protein directly to where it's needed in the body, aiming to restore the cellular function by reducing this substrate, these lipids that are accumulated in the cells of these Fabry patients. This strategy, which is based on the intravenous administration of an exogenous α-galactosidase, which is encapsulated in the nanoliposomes, protects the protein from degradation and permits the delivery of the GLA selectively in target tissues, because also the surface of these nanovesicles or these nanoliposomes are functionalized with a targeting ligand that has a high affinity for proteins that are overexpressed in these affected cells in the Fabry disease. This can make the treatment safer and more effective compared to current enzyme replacement therapies. So this new nanoformulation that is under development, we have named this as NanoGLA. And as mentioned, is based on nanoliposomes, but encapsulate a new recombinant human α-galactosidase that is produced by the CMO Leanbio. And for the manufacturing of this nanoformulation, we use the DELOS technology, which is a robust and green manufacturing process developed by the Institute of Material Science researchers and scale up and currently owned by Nanomol Technologies.
Alba, can you explain a little bit more about the DELOS technology mentioned?
As Elizabeth mentioned, so DELOS is a nanoparticle manufacturing technology, which is patent protected, and it's currently owned exclusively exploited by Nanomol Technologies. And its advantage, it is a single-step manufacturing procedure that enables us to obtain, for example, this nanoliposome, entrapping the GLA enzyme, and also functionalize with a peptide and controlling very well the particle properties of the formulation. So this means that we are obtaining this formulation with controlled particle size, controlled particle morphology, controlled stability, and it enables to have a very good quality at the end. So this is like the key advantage.
Okay. Ellie, you mentioned a little bit about the seriousness of Fabry disease. Can you tell us more about your motivation for this demo case and how you got on the idea of looking for a treatment?
Sure. Our motivation was to develop a more effective enzyme replacement therapy for patients with Fabry disease. Current treatments have limitations like fast degradation of the enzyme, poor distribution in the body, and importantly, the inability to cross the blood-brain barrier to reach the brain. So this is something that we want to overcome with this new nanomedicine. And in the frame of PHOENIX project, our goal was to move this new nanomedicine to an advanced stage of pharmaceutical development closer to clinical application.
Okay. And what were you hoping that, or are you hoping that the impact will be, or have you seen some impact so far?
So bringing this nanomedicine to market would allow us to offer a more effective enzyme replacement therapy based on nanotechnology. This would open new treatment options for a rare disease, improving both the quality of life and life expectancy of patients. This is a future perspective.
Great. And Alba, from your side?
So maybe a future perspective is that also thanks to PHOENIX, we were able to scale up the technology and also set up a GMP manufacturing plant. So what this will imply is that we will be able to supply this material for clinical testing, which it was not possible before. So it will help us also, as Elizabeth mentions, to bring this therapy closer to the patients, and also why not to perform like similar approaches for related diseases.
That's great. The project is coming to a close soon. Can you tell us about what results have been achieved so far?
So far, the results are very promising. In preclinical models, NanoGLA show better efficacy than current approved therapies that use the GLA in the free form, non-nanoformulated. So the NanoGLA has demonstrated superior efficacy in affected organs, including the brain, which is especially difficult to reach and still not addressed by approved enzyme replacement therapies. The results highlight the potential of the NanoGLA to treat both the systemic and brain-related symptoms of Fabry disease. And this also was recognized in 2021 by the European Medicines Agency, this significant benefit that the NanoGLA can offer over current enzyme replacement therapies, granting the Orphan Drug designation. And this encouraged to advance this nanoformulation toward clinical translation. Due to these interesting results, we have founded a spin-off named Leanbio Pharmaceuticals. We have found, together with researchers from our group, from the Institute of Material Science of Barcelona, together with Nanomol Technologies, and with the objective to advance the pharmaceutical development of the NanoGLA to the clinical phase, and in addition to expand the application of this encapsulation platform to other nanomedicines for rare disease.
That's really exciting. Congratulations. So within the PHOENIX project, you were able to work on this demo case, accessing on one hand a variety of services and bringing together the three of your companies, but also with this single entry point, which has been developed during PHOENIX. So what did that mean? How did it make a difference to be accessing these services through a single entry point and through the project itself?
So having access to PHOENIX services has been a great opportunity to continue with the development of the NanoGLA. First, the funding from the European Commission gave us essential support to keep progressing. We were able to develop most of the physicochemical and biological methods for the characterization and quality control of the nanoformulation. PHOENIX also connected us with expert partners who provided specialized services, like, for example, for the characterization of the surface of the properties of this nanoformulation, or for lyophilization and in vitro and vivo testing. Also, Leanbio could scale up the production of the GLA enzyme in order to manufacture this in the quality and enough quantity for the preclinical testing. And also from the Nanomol Technologies side, Alba can contribute with the significant milestone achieved here in the manufacturing process.
Yes, thank you, Elizabeth. So yes, from the CMC side, I think the main benefit that PHOENIX has given to the NanoGLA demo case is the possibility of having a GMP compliant equipment that has been set up and has been successfully used to manufacture big batch of NanoGLA of almost 30 liters. So this is a huge amount for us compared to the milliliter scale, no? That was at the beginning of the project. And and this has been specifically tailored to the unique nanoparticle technology that we use. So this has been very, very helpful and very great for the demo case. And besides that, also it allowed us also to implement a GMP compliant HPLC technique. So we have also developed expertise on analytical development of nanomedicine that is important in order to release the batches that are used in the different stages and progressing further to the clinical stage. So yes, this from the CMC part, I think it's the main goals achieved.
Okay. And for other companies that are, or research groups that are listening and think that this sounds interesting, who do you think can benefit the most by accessing PHOENIX services from a single entry point?
So a single entry point like PHOENIX is indeed a great opportunity because it brings together all the services and expertise in one place. This permits to save time, reduce stress, and helps researchers focus on what is important, advancing their innovations. And also for complex areas like nanopharmaceuticals, that having all this coordinated support makes a big difference. And at the end, it helps to make this journey more efficient.
Yeah. Well, thank you so much for sharing about your experience working with PHOENIX, about your demo case, and we look forward to seeing your formulation brought to the market in the future.